A decade ago, an exciting journey began with the creation of the Kidney Health Initiative. KEY became a vehicle for the kidney community to foster innovation while ensuring patient safety. A voice was given to patients, empowering them to influence clinical trials and new treatments. As membership grew, KEY mapped a vision for improved patient quality of life, even as we faced unprecedented challenges. Still, we continued to push forward, building momentum for biomarker development and fueling alternatives to conventional dialysis.
For over a decade, we've worked together to spark innovation and develop safe and effective patient-centered therapies for people with kidney diseases.
That is the mission of the Kidney Health Initiative. And the progress we've made together has been impressive, and we've made real, tangible differences.
But really, this is just the beginning, and we're excited to keep working together and making progress that will impact those battling kidney disease.
Please welcome ASN counselor, Dr. Linda Freed.
Good morning. My name is Linda Freed, and I currently serve as an ASN counselor. And it's my pleasure at this time to acknowledge two outstanding individuals for their time, talent, and leadership to our journals. Dr. Josie Briggs has served as editor-in-chief of the Journal American Society of Nephrology for 6 years and has added tremendous value to the Society's mission and expanded the knowledge of countless kidney professionals around the world. Before I joined as counselor, I had the pleasure of serving as associate editor under Josie, and I want to say she was a great editor as well for those of us who are AEs. Established in 1990, JASN is the flagship journal for ASN. During Dr. Briggs' tenure, JASN published over 1,000 original research articles, approximately 47% in basic research, with the remaining covering clinical epidemiology, clinical research, meta-analysis, and rapid communication. Her editorial team never wavered in their dedication to JASN and the editorial process during the uncertainty of COVID, maintaining the highest standards throughout the peer review process, despite global challenges of the pandemic. Under Dr. Briggs' leadership, the JASN editorial team also launched the JASN Editorial Fellowship Program in 2019, matching early career scientists, junior faculty, or senior trainees with a JASN associate or deputy editor for a 2-year mentorship program. And over 30 editorial fellows have already participated in this invaluable program. Overseeing a journal of JASN's caliber requires continuous, unceasing effort over many years, and ASN is extremely grateful to Dr. Briggs for her unyielding dedication and commitment. Dr. Rajneesh Mehrotra has served an unprecedented 7-year term as editor-in-chief of the Clinical Journal of the American Society of Nephrology. Several changes to ASN's journal publishing program, including a partnership with Wolters Kluwer and an upcoming move to a more holistic ASN journal portfolio structure in 2024, led the ASN Publications Committee and Council to request that he extend his editorship for an additional year. ASN is extremely grateful to Dr. Mehrotra and his entire editorial team for dedicating an additional year of service to CJASN. During his term, CJASN expanded the popular special series and produced nearly 100 individual special series articles across 6 themed topics. These articles have now been downloaded nearly 2 million times and cited over 4,500 times. Furthermore, Dr. Mehrotra's editorial team introduced patient voice articles, podcasts, and 2 training programs. Overseeing a journal operation like CJASN's requires a constant commitment to producing high-quality, novel research that moves the nephrology field forward time and again. Dr. Mehrotra and his team excelled in doing this for 7 years in 84 CJASN issues. ASN Council fully recognizes the considerable obligations involved with JASN and CJASN, and we are most grateful for all that Dr. Briggs and Dr. Mehrotra have done to advance the Society, our journals, and the kidney community. And with that, I'd like to invite them to come up to the stage.
Well, it's my turn to express a few thanks. It's been a really important 5-6 years for all of us, and I've benefited from a superb team, and I want to thank the people who have made our work possible. It's a long list, and I'm not going to read the entire list, but I want to specifically mention Mark Knepper, who has been our editor for all basic science work, and first Paul Polewski and Don Wesson, who've carried the burden of looking at being the first person to look at the clinical work. We've also had enormous support from Tom Hostetter, who's given us wisdom and history, and from Joanne Bargman, who has handled a lot of the COVID work, as well as all the letters to the editor. And finally, the additional deputy is Neil Poe, who has a view of big issues and has served as our guide on many of the equity matters. We've also been incredibly well supported by ASN staff, especially Sydney Koff and Bonnie O'Brien. So thank you. I am the one to thank.
It's a moment for me to express gratitude also to a whole host of people. I want to start by thanking the ASN and the ASN Publications Committee. When I was an applicant for this role, it was not obvious that I was the best candidate for the job. They entrusted me with this responsibility, and any success that we've had is because of the team that we've had. Most importantly, I want to thank Dr. DeBoer and Dr. Chancho. They were the two deputy editors. They worked at my side and have always been available for their wise counsel, but also the entire group of editorial associate editors, the authors that submitted the papers to us, specifically also the patient authors and the patient editors that have now submitted 84 articles to the pages of CJASN, which is a powerful voice from the patient community. I want to thank the reviewers. I want to thank the reviewers, the visual abstract editors, the ASN staff. I want to thank particularly Sherry and Natalie, but also Susan and Kim, who've worked behind the scenes to make things go on. And finally, for Bill Bennett and Gary Carhan that established the journal. And I am thrilled to pass this on to Dr. Rhee as the incoming next editor-in-chief for CJASN. Thank you.
Right. Good morning. My name's Sue Quaggan, and it has been my privilege to serve ASN Council for 8 years and to serve as this year's chair of ASN's Awards Selection Committee. The John Peters Award was established in 1983 to recognize substantial achievements in research in the field of nephrology, as well as sustained achievements in one or more of the following: clinical care, research, and leadership.
John Peters was clearly one of a kind.
He was groundbreaking.
He believed in progress, and he believed progress needed pushing.
John Peters really is considered sort of the father of modern nephrology. He really was instrumental in quantifying measurements of things like urine and blood.
His laboratory was the bedside. He did his research at the bedside trying to understand patients.
Dr. Peters really was a leader in using actual measurements in patients to uncover pathophysiology and to apply the advances in basic physiology to actually see how that could benefit patient care in the real world.
Quantitative Clinical Chemistry was a groundbreaking book. It forced everybody to think in theorems and then test these theorems by doing interesting experiments.
He was sort of an early translational scientist. I think Peters was ahead of his time, and I think he was a visionary.
Peters wanted to speed up the evolutionary process of medical progress through medical science and education with federal funding.
My grandfather had very strong values and felt it was very important that medical care be given to all Americans.
It's interesting to think about current debate and recent healthcare legislation in light of what Peters promoted and pushed for back in the 1930s.
Amidst all of his medical excellence, he was committed to healthcare.
It's this kind of breadth of concern for the health of the entire healthcare system that made John Peters unique and established the legacy for the wonderful recipients we've had for the John Peters Award.
The American Society of Nephrology presents the 2023 John P. Peters Award to Dr. Catherine Tuttle for her substantial contributions to nephrology research and academic medicine. She is Executive Director for Research at Providence Inland Northwest Health, Regional Principal Investigator of the Institute of Translational Health Sciences, and Professor of Medicine at the University of Washington. She oversees a regional network of 17 clinical research centers and chairs the Research Regional Executive Council for UW. A native of the Northwest, Dr. Tuttle has spent most of her career in Eastern Washington. She earned her medical degree and completed her residency in internal medicine at Northwestern University in Chicago. She was a fellow in metabolism and endocrinology at Washington University in St. Louis and completed her nephrology fellowship at the University of Texas Health Science Center in San Antonio. Dr. Tuttle's major research interests are in diabetes and chronic kidney disease. She has published over 320 peer-reviewed articles, including an early landmark study elucidating physiological principles underlying glomerular hyperfiltration in humans with diabetes. This led to a number of studies that laid a foundation for new therapeutic targets and clinical trials. Her work has helped establish SGLT2 inhibition as the most impactful therapy to date to reduce risks of kidney failure, cardiovascular events, and death in persons with and without diabetes. In addition to her teaching responsibilities at the University of Washington, Dr. Tuttle has participated in numerous CME courses locally, nationally, and internationally. She lectures frequently across the globe to optimize the care of patients with diabetes and kidney disease, and continually advocates for equitable care, particularly for minority patients. She leads the Cure CKD registry of real-world data for chronic kidney disease, diabetes, pre-diabetes, and hypertension from nearly 4 million patients. Dr. Tuttle has served a number of national and international organizations. Currently chair of ASN's Diabetic Kidney Disease Collaborative, she was on the inaugural board of directors for the Kidney Health Initiative and has chaired numerous working groups and committees for ASN, the NIH, and the National Kidney Foundation, among others. Dr. Tuttle and her husband, Dr. Andrew Boulay, reside in Spokane, Washington. They and their daughter Angela enjoy skiing and other outdoor activities. The recipient of many awards and honors throughout her distinguished career, Dr. Catherine Tuttle is a stalwart leader, mentor, and role model to many, especially women, in the field of nephrology.
So I am thrilled to present this year's John Peters Award to none other than Dr. Catherine Tuttle.
Thank you. I'm truly humbled in this moment. I'm very grateful to the American Society of Nephrology, the Awards Committee, and Women in Nephrology for considering me worthy of the John P. Peters Award. Neither of my parents had a four-year college education. But they told me if I worked hard and did well in school that I could have one, and that education would be my ticket in life. And they were so right. I went to the University of California, Irvine, where I studied art and science, and then went on to Northwestern in Chicago. That's where I found my passion for all things metabolism and physiology. Early mentors there in endocrinology, Mark Malich, and nephrology, Francesco Del Greco, and Murray Levin encouraged me to keep going further. At my next stop in St. Louis, Washington University, where I did my fellowship in endocrinology and metabolism under the tutelage of Phil Cryer, I learned the techniques of insulin and glucose clamping that were used to unravel mysteries of glucose homeostasis. Our study participants were mostly younger people with type 1 diabetes who endured day-long experiments on the artificial pancreas, a biostator that filled an entire room. They endured these experiments because they truly believed that somehow, some way, that the science would make a difference for them. Yet they were melting in front of our eyes from severe complications, particularly kidney disease, which went largely unrecognized until it was a devastating event in their lives. And that's when I felt the calling, my calling, to make diabetes and kidney disease my mission. I went on to the University of Texas Health Science Center, San Antonio, and there physiology was king. And I learned from some of the best, Bob Kunow, Jay Stein, Dwight McKinney, and Meyer Lifshitz. They were doing micropuncture studies to disentangle the mysteries of glomerular hemodynamics and tubular transport process. Upon the arrival of Dr. Ralph DeFranzo, we teamed up with our common interests in the kidney and metabolism to translate learnings from the rat models to humans, and using our techniques of insulin and glucose clamping to do them under very controlled metabolic conditions. We actually discovered that persons with both type 1 and type 2 diabetes actually had an augmented hyperperfusion and hyperfiltration response to stimuli, such as amino acids and glucagon. And I will always remember the day in 1987 when, in the bench to bedside and back ethos of the time, Ralph hatched a plan to give Florazin to diabetic rats. The rest is history. I returned to Eastern Washington and spent several years in clinical practice. But research called me back because there were such enormous unmet needs to improve the treatment of people with diabetes and kidney disease. I was given an opportunity to lead the research program at our healthcare system, now known as Providence, which had been the home to the academic programs at the University of Washington for decades. We reestablished the physiology lab and then turned our attention to cellular and molecular biology because we wanted to take a deeper dive into the way kidney damage was occurring in people with diabetes. Our focus was on aberrant metabolic products and activation of innate immune responses. Several of these discoveries have since been translated into new biomarkers and clinical treatments as well. I want to give many thanks to my colleagues and mentors at the University of Washington. In the early days, Bill Kauser, Donald Sherrard, Rick Johnson, Charlie Alpers, and Stuart Shanklin, and then later with the establishment of the Clinical Research Institute, Jonathan Himmelfarb, Ian DeBoer, Raj Mehrotra, and Mary Mafkarian. With the advent of the electronic health record, we established the Cure CKD registry, which now contains over 4 million unique individuals with kidney disease or at high risk by virtue of diabetes or hypertension. This has been a long-standing and enduring partnership between Providence and UCLA Health, especially Keith Norris and Suzanne Nicholas. However, we were both surprised and sobered by the early reports out of Cure CKD, showing the marked under-recognition and under-treatment of kidney disease in contemporary U.S. healthcare systems, now corroborated by many other reports. These data herald a call to action, that we must move much more expeditiously from evidence generation to implementation. Our patients deserve better, and they can only benefit from the treatments they receive. I'd like to thank my Spokane teams in the laboratory, Rick Meek, Cheryl Cooney, Bob Anderberg, and Brad Dieter, clinical research, Raditza Alisic, Josh Newmiller, and Michael Ryan, and our wonderful biostats and methodology core led by Bob Short, Kandaratha, and Cammie Jones. I could not have done it without you. I'd also like to thank my family, my husband Andrew and Angela, who are here today, who put up with my many long days at work and nights away while keeping the magic aglow at home. I'm also joined by my sister Maureen Postrino and her family and friends who are the family we choose. Thank you for being here. And finally, big thanks to all of you for the good work you do every day. I look forward to the journey ahead as we now move to a vision of kidney health for all, with the powerful tools now in hand to save lives, hearts, and kidneys. Thank you.
The American Society of Nephrology and the Council on the Kidney and Cardiovascular Disease of the American Heart Association are proud to present the Donald Seldin Young Investigator Award. Dr. Seldin was a giant in the field of nephrology and helped to create the American Society of Nephrology, serving as the Society's second president.
Young investigators in kidney disease are definitely a hope for the future.
The young investigator often has a new approach.
Don Seldin was a master teacher and clinician.
He had tremendous impact on young investigators.
One of the major advantages to the whole community of the Young Investigator Award is to highlight new areas of science.
We believe that funding young investigators is the key to keeping the next generation of scientists bringing new treatments for both kidney disease and heart disease.
Dr. Seldin always encouraged young investigators to go after with dedication and diligence. Give it everything you've got. He was a leader in academic nephrology, not only in research, but also in the practice of nephrology and education.
The Department of Medicine at Southwestern took giant steps forward because of Dr. Seldin. He helped many young investigators by directing them to the most logical set of experiments that would dig into the problem they were studying.
He paid a lot of attention to young investigators because he realizes they are the future.
Young investigators are actually making new discoveries that will help everyone, not only in nephrology, but in all fields of research.
Research is at a really exciting time. We know more, we have more technology than we ever had before, so the potential to impact human disease is great.
I think it's appropriate for Dr. Seldin's name to be wedded to the Young Investigator Award. It bonds his name to those individuals who will carry science forward into a new era.
The 2023 Donald W. Seldin Young Investigator Award, co-sponsored by the American Society of Nephrology and the American Heart Association's Council on the Kidney and Cardiovascular Disease, is presented to Dr. Benjamin Bino Friedman for his seminal discoveries in basic and translational kidney stem cell research. Currently an associate professor in the University of Washington's Division of Nephrology, he is also an investigator at the Kidney Research Institute and a core faculty member of the Institute for Stem Cell and Regenerative Medicine. After earning his PhD in molecular and cell biology at the University of California, Berkeley, Dr. Friedman became interested in nephrology as a postgraduate fellow. He joined the lab of Dr. Joseph Bonventry at Brigham and Women's Hospital and Harvard Medical School, first as a fellow and then as a faculty member. These training experiences paved the way for his recruitment to UW in 2015, where he has received mentorship from Dr. Jonathan Himmelfarb. Dr. Friedman pioneered the first techniques to differentiate human pluripotent stem cells into kidney organoids. he demonstrated that sophisticated phenotypes of kidney disease can be reconstituted in organoids to reveal novel disease mechanisms. As a very early adopter of CRISPR genome editing to model kidney disease and therapy, the Friedman Laboratory has played a critical role in integrating this new technology into the nephrology world. Dr. Friedman was the first to leverage the power of automation to mass-produce organoids from stem cells with liquid-handling robots, which led to improved organoid vascularization and the identification of a new molecular pathway involved in polycystic kidney disease. He has long expressed interest in developing kidney regeneration as a therapeutic area and has shown that human kidney organoids can successfully form grafts when implanted into mice. Dr. Friedman's original research articles have been published in many leading scientific journals, including Nature Materials, Cell Stem Cell, and JASN. He has authored a series of invited reviews and commentaries, as well as 5 book chapters. His work has also been featured in popular press outlets such as GeekWire and Cairo 7 TV News.
With the help from robots, he grows kidneys.
He has delivered numerous invited lectures at national and international conferences. Dr. Friedman's work has been recognized with many prestigious honors, including the Stem Cells Young Investigator Award and the UW Medicine Fialco Scholars Award. On the home front, Dr. Friedman enjoys spending time with his wife Hangsha, their son Bruce, and the extended Friedman family. With so many noteworthy accomplishments at a young age, Dr. Bino Friedman is an inspiration to other young investigators and the entire nephrology community.
I am thrilled to announce that this year's Donald W. Seldin Young Investigator Award is awarded to Dr. Benjamin Bino Friedman, and he will present his work today on human kidney organoids for disease modeling and regeneration.
Thank you so much. This is so meaningful to me. Thank you to the ASN and to the AHA. The ASN has always felt like home for me. And that just makes this extra sweet to be here today and receive this tremendous honor and to be able to take this honor in the name of Dr. Seldin. So thank you. I also wanted to take a moment just to recognize all the amazing young investigators that are part of our community. Let's take a moment and applaud all of them. I really am accepting this award in their honor as well. And part of the award is also for the organoids, whose story I will now tell. We live in a remarkable time, and it's really for the first time in human history, we're able not just to study organ development, but to recreate it from our own cells. And this is how it works. We can culture cells, any type of cell from our own bodies. These are adult cells. They're not stem cells. But by introducing into them stem cell genes, we can reprogram them to a very early stage of development before any of the organs have formed, where they're pluripotent. And these are what we call the induced pluripotent stem cells, or iPS cells. And these can then be changed or differentiated into organ lineages and form these tissue-like structures called organoids, which we can study in the dish or put back into the original patient where they would be 100% immunocompatible and be used as a transplant. And for me, this is a personal technology as well. This picture is of my uncle, shown here, with his family. He's had 2 kidney transplants. And although these have sustained his life, over the decades, the constant immunosuppression and visits to the hospital have taken their tolls. And this got me thinking maybe we could use iPS cells to help people like my uncle grow new nephrons of his own. So the trouble was that when we started, iPS cells had been differentiated into heart and liver and other lineages, but never kidney. So as a postdoc, I took on this challenge and I made a breakthrough, and I discovered a way to change iPS cells into the kidney lineage. And this turned out to be worth the wait. So you can see what this looks like over a series of a few weeks in this movie, and the stem cells change into a mesenchyme, and then they epithelialize before our eyes into these tubular condensates. And when we look at these condensates by microscopy, we can identify podocytes, shown here in red, proximal tubules in green, and other types of epithelial cells found in the kidney. And these are taking on structures that are very similar to kidney tissue. It's not perfect, but it's remarkable to appreciate that the structure shown here on the left was once a skin cell and has now totally changed its fate and turned into a kidney-like organoid. And we now appreciate that organoids, kidney organoids, are nephron-like in many ways. Not only do they have many different cell types that we associate with the kidney nephrons, but these also adopt morphologies that are representative of the complex structures we see in vivo. In addition to the epithelial cells, there's also stromal cells and endothelial cells, which are interacting with the epithelia in ways that approximate what happens in our bodies. And these structures can perform primitive functions that we would associate with the kidney, including glucose transport. So today I'm going to give you a talk in 3 parts. First, I'm going to discuss our efforts to model disease in a dish. Then I'm going to talk about our progress towards regenerative medicine. And finally, I'm going to step back and present what I call the bigger picture, my life as a stem cell researcher. And the immediate application of these organoids is to study disease and help us understand both how the diseases work and how we could potentially intervene with them. And to start, we focused on polycystic kidney disease. This is a fascinating disorder, affects millions of people worldwide, in which the tiny tubes within our kidneys swell up and form these balloon-like cysts. And we know that PKD is caused by mutations in either of 2 genes, one encoding polycystin 1, the other encoding polycystin 2, and that these form a channel receptor complex at the membrane of the cell. And we know that the cysts form from the tubules, but we don't understand well how this actually occurs. And one of the barriers in the field is that the simple cells that we use in vitro don't form cysts in a PKD-specific way. So we didn't have an in vitro model. So to create one, we used what at the time was a relatively new technology called gene editing. And this uses an enzyme to cut DNA and introduce mutations at specific sites. And using this, we created iPS cells with PKD knockout mutations in either PC1 or PC2, and identical twin cells, isogenic controls, which did not have those mutations. And we differentiated them into organoids. And we observed something remarkable. Initially, the organoids formed normally, but then specifically in the organoids with the PKD mutations, we saw these little bubble structures arising from the tubules. And these were epithelial cells that stretched out and filled with fluid. And it was very specific to the disease mutants and not their identical twin controls. And this was essentially PKD in a dish. And it taught us that PKD cyst formation is a fundamental process that we can recreate with cells in a petri dish and now study and try to understand. And these cysts can get very big. This is a movie on the left, And you can see these started off as organoids that were 1/10 of a millimeter in diameter, and they've now swollen to sizes of centimeters. They're filled with fluid, which is a different color from the surrounding media because they're transporting out the phenol red dye indicator. And this can be very efficient when the cells are placed in a suspension culture, where we're getting now 80% cyst formation in our PKD null lines, and it's still very low levels in the controls. Now, one of the beauties of the organoids is you can grow them in many different formats in diverse ways, and we've taken advantage of this. We've grown organoids in miniaturized formats, 384 well plates, and you can see each little tiny well here has a smattering of kidney-like structures. And we can now treat each individual well with a different drug at a different dose and assess its impact on polycystic kidney disease. And this has led us to an appreciation of a new pathway, myosin, which we believe is involved in this disorder. In parallel, we've taken organoids and investigated what is the effect of flow on this phenotype by placing them in microfluidic chip devices. And what we observe is that as fluid flows over them, the organoids swell up and form the cysts grow larger, specific to PKD. And this is implicating absorption in the process. And this has led to novel hypotheses for how PKD is actually occurring. And what we believe is happening now in the organoids is that glucose is being absorbed by apical surface of the epithelial cells, and this is driving water uptake into the organoids. This causes the epithelium to stretch and distend as they fill with fluid. And this is a very different type of process than we would originally have imagined is occurring, but this is what the organoids are telling us. And it's leading us to new therapeutic strategies for PKD, such as inhibiting glucose absorption or activating myosin to stiffen the structures and prevent their distension. These PKD organoids are being sought after by drug development companies to understand whether their therapeutics could be repurposed for polycystic kidney disease. And this is one story where we've now generated a new cohort of mutants using CRISPR base editing just to change a single base pair in the DNA and cause truncating mutations in either PKD1 or PKD2. And now we can apply a novel class of drugs called read-through therapeutics to enable the ribosomes to essentially ignore the stop sign and create full-length polycystin, as shown here. And this actually works. And as we titer in this read-through therapeutic compound, we see that the cysts get smaller. And this is not due to cytotoxicity because the structures are still healthy under these conditions. Sharday Thomas, who's a research scientist in our laboratory, has a poster describing this novel cohort today, so I would encourage you to check it out. It's not just PKD. Models are emerging for many different renal disorders. We've got a model of APOL1 disease, and Dr. Ben Juliar, who's a postdoc in our lab, has a talk about that tomorrow. There are ciliopathies, which form cysts similar to PKD, hints of diabetic kidney disease, infection with SARS-CoV-2 virus in the proximal tubules, and glomerulosclerosis models. And in each of these cases, we're seeing an interplay between genes and the environment that lead to the disease. So the conclusions for this first part of my talk, I think all models are wrong, but some are useful. Phenotypic models emerging from our organoids are revealing key ingredients of disease in ways that we would not imagine by ourselves. And these human organoids constitute novel drug development tools. And this is just in time for the FDA Modernization Act, which allows an applicant for market approval for a new drug to use methods other than animal testing, including organoids. And I see this integrating at different stages of the drug development pipeline, and hopefully this will lead to a greater number of approvals by authorities such as the FDA. And faster as well, because we need new treatments. So now I'd like to talk a little bit about the applications of regenerative medicine for the organoids. And we're doing this through the prism of a patient population, which is the patients with cystinosis. And this is a disorder in which there's a mutation in a lysosomal transporter for cysteine, the amino acid. And this causes accumulation of cysteine in these cells, and they become unhealthy and sick over time. And for reasons that we don't fully understand, the kidneys are particularly vulnerable to the effects of cystinosis. And this leads to a requirement for kidney transplantation early in life for these patients. So we've come up with a plan to make tissues cystinosis-free. We're going to start by making stem cells from the patients. Then we're going to introduce into them gene therapy to prevent the disease from recurring. And finally, we're going to transplant that back into that patient themselves. And as a starting point, we've developed a biobank of iPS cells from patients with cystinosis. And the way we do this is by collecting urinary cells. We send out a kit to the patients by mail, and they provide us a large urine sample. And we can derive these urinary cells by retrieving that back to our lab. And then we can reprogram them into iPS cells and subsequently kidney organoids. And we've done this from 18 patients with cystinosis. Next, we add back cystinosin, and to do this, we've developed a gene cassette which targets, using CRISPR-Cas9, the safe harbor locus of our DNA. This is a special spot within the DNA that we can essentially copy or paste genes. And in this case, we're knocking in, not knocking out. So we're adding back a copy of cystinosin, and this is a doxycycline-inducible copy, so that if we add doxycycline, it turns on, and you can see this here. Otherwise, it's turned off, so that we have a very elegant internal control with the non-doxycycline treated. And you can see here that adding this gene back into the cells actually reduces the levels of cysteine accumulation, which is the pathognomonic cause of the disease. So finally, we're implanting these back into not patients, but animals. And that's important because we wouldn't want to start with patients here. You can see here how we deliver the stem cells beneath the capsule of a kidney. And this is an immunodeficient mouse. And the mice are fine after this. And the graft then grows in the animal for weeks. And when we then examine the graft, we've observed something remarkable. We see that endothelial cells from the mouse, these vascular cells, are climbing into the graft. And they're vascularizing the podocytes, the human podocytes, shown here in red, to form what we call glomerulus-like structures. And we've never seen this type of vascularization in a dish, but somehow, by growing the organoids in a living being, they know how to develop glomeruli. And these glomeruli are also better organized and much more sophisticated than what we get in the dish. We see cell populations, not just podocytes, but parietal epithelial cells, and even mesangial cells, the stromal populations of the glomerulus, are present. So the second conclusion I'm going to draw is that a humble urine sample can be the start of something big. We've developed a new model of nephropathic cystinosis with gene therapy, and we've shown that grafts contain chimeric glomerulus-like structures, which are the first hints towards function, which is the holy grail of the field. And this suggests a pathway whereby we might be able to start iPS cells and gene editing as a new area of kidney transplant, and that's very exciting. And to really develop this further, we're now developing immunodeficient mouse models with disease mutations that we can use to simulate the patient population and make sure these types of applications would be safe as well as efficacious before trying them out in people. So the last few minutes, I just want to step back and discuss what I call the bigger picture, some of the lessons that I've learned as a stem cell biologist. The first lesson that I'd like to share is that people are more important than cells. I take this for granted. This would normally be the part of the talk where I kind of give my acknowledgement slide and thank the people who have actually done the work. And I'm showing you them here. And this is really a tremendous group of individuals, and it's been my great privilege to work with them. You know, science is really coming from people, and working with my students and postdocs and research scientists has just taught me so much. And I think of them as pluripotent stem cells in a certain way. They're at the pluripotent stage. They can turn into anything, become anything they want, and that's what's so exciting. But they're also delicate, just like the stem cells. They need nurturing. They need attention every day. And this is sometimes a struggle. It's sometimes a challenge for everybody. And we live in an age where students they're getting a lot of challenges. Science, to some degree, is under attack. And we have to address this, and we have to change how we do things to keep up with the times. So how can we help students? I think we need specific educational programs for folks like the people in my lab who want to contribute to medicine, but they're doing it from a basic research side. And we need to tailor these programs. We can't just rely on the old style programs that have been in existence for a long time. We need models for scalable lab growth and to give each individual the attention that they need. We need to remind our students of the fun and joy that's inherent in what we're doing as scientists, why we got into it in the first place. And we need to establish positive mentoring relationships so that they can see that joy in what we're doing today. I want to give credit to my parents, my father, Mark, who's a physician, and my mother, Linda, who's a marital therapist. They're both here today. My father really gave me the genes and the nurturing for the science part of my life. But my mother gave me the attention to the mental health aspect. And I think that's equally important. So thank you. Thank you, parents. I know this to be true because I was once a student not so long ago. This is a picture of me from when I was at Penn, just a mile or so away from here, in Dr. Ellen Heberkatz's lab, who's also here today. And Ellen took me in and gave me so much freedom to explore. I really got that fun and joy of science at such a formative age. Thank you, Ellen. And subsequently, I went on to Berkeley to Rebecca Heald's lab, and Rebecca taught me to think big and to follow my dreams. But most of all, she taught me to be kind. And if I can paraphrase Rebecca, I would say, if life is a stem cell, then love is the organoid. Now, if students are our pluripotent stem cells, then I think the young PIs must be somewhere around the renal vesicle stage. We're not quite fully differentiated yet. We've committed to a path, and we're excited about it. But we also, as anyone in our lab will tell you, that stage is a very sensitive stage of development. And although I've enjoyed my measure of success, I am very well aware of the pressures of being a young PI, and sometimes I even feel a little bit like an outlier on a Manhattan plot. So how can we help young PIs? I think we need to find more defined and sustained support mechanisms and job security. This is true both for scientists as well as for clinicians. We need to really think about our appointments a little bit more carefully and design them in ways where PIs can feel comfortable and free to explore. We need investment in discovery and translation that lets us chase down our dreams and gives us the freedom to operate. You know, it's wonderful to build big buildings on college campuses. But those buildings are useless if we're not giving equal attention and investment into the people who occupy them. We need to bolster and support the advisor-student relationship in a thoughtful and meaningful way. And when things get tough, we need to provide love and trust for those young mentors. And things do get tough, as we've all experienced during COVID. And we should think to ourselves, how supportive were we during that time for our young investigators? I'm very fortunate. I have this great family to go home to, and this is my wife, Hangsha, who's an assistant professor, so we can commiserate and calm each other down when we run into any sort of troubles. And my son, Bruce, who just holds my hand and makes me feel better about everything. So thank you to you guys as well. If we do this, if we establish these types of growth niches, then it's going to enable research breakthroughs. And I've been fortunate to be part of 2 such niches. Most recently at the University of Washington, under the guidance of Stuart Shanklin and Jonathan Himmelfarb, they gave me the ability to do all the research that I described today and gave me that team. So thank you, Stuart and Jonathan. And then previously at the Harvard Medical School, I had a wonderful trio of mentors, Joe Bonventry, Jing Zhou, and Ted Steinman, as well as my colleague and peer, Albert Lamb, who taught me how to culture stem cells and worked with me side by side during those disease modeling and differentiation early days. So thank you to all of you as well. I've had a wonderful division supporting me and a large host of collaborators, both within the US and worldwide. And this has given me the environment that I needed, the microenvironment that I needed to succeed. And this is just some of the names. So finally, I wanted to thank our funders. And this is a combination of government, foundation, and private that has supported this work and enabled it to develop to the stage that it's at. I wanted to thank my lab once again and give a special shout out to patients who are taking control of their own destiny and contributing both in funds and time and cells to our work and to hope for their own futures. So thank you.
Greetings to you all. I'm Diane McKay, and as co-chair of the Kidney Week Committee, along with my colleague and friend Dr. Mark Parazella, it's really been a privilege to be involved in orchestrating a meeting of such magnitude. Today, however, stands out as a particularly special moment, as I have the honor of introducing one of our eminent plenary speakers. Delving into the world of sensory perceptions, our distinguished speaker from the Scripps Research Institute in La Jolla, California, is Dr. Arden Patapoutian. His enlightening lecture today is titled, "How Do You Feel? The Molecules That Sense Touch." At the forefront of groundbreaking research, Dr. Patapoutian's laboratory unveiled the pivotal molecules involved in our sensations of touch and pain. This monumental discovery was fittingly recognized as he was awarded the Nobel Prize in Physiology or Medicine in 2021. Hailing originally from Lebanon, Dr. Patapoutian embarked on his academic journey in the United States in 1986. He completed undergraduate studies at the University of California, Los Angeles, and pursued his doctorate degree at the California Institute of Technology. He joined the Scripps Research faculty in 2000, where he's now the Presidential Endowed Chair in Neurobiology and serves as an investigator for the Howard Hughes Medical Institute. With memberships in the National Academy of Sciences and the American Academy of Arts and Sciences, Dr. Padapushin's accolades are numerous, including being co-recipient of the 2020 Kavali Prize in Neuroscience and the recipient of both the 2021 BBVA Foundation Frontiers of Knowledge Award and, of course, the Nobel Prize. Ladies and gentlemen, it's with immense respect and anticipation that I welcome Dr. Adam Padapushin.
Good morning, everybody. Thank you for the invitation. So this is my favorite photograph that kind of tells me how important the sense of touch is. This is the White House, and this little boy comes and tells the president that other kids tell him that his hair looks like the president. And Barack Obama does not miss a beat and bends over and says, "Touch it." And so we're not just talking about this child recognizing the texture of the hair, but that intimate nature of what we call affective touch, recognizing that someone that not just looks like him, but the hair feels the same is the President of the United States. In addition to the sense of touch, what you will notice if you look closely is that the President's eyes are closed, and the fact that he can bend over like that and not fall down is due to one of the most important senses that you have that many of you probably don't know about it. It's called proprioception. It's sensory neurons that innervate muscle, and from how much those muscles are stretched, you know exactly where your limbs are in space. That's how you can close your eyes and touch your nose, for example. We'll talk about all of this today. So all of these senses start in a group of neurons we call dorsal root ganglia. They're just dorsal to the spinal cord, as shown here. And one of the major characteristics is that they're extremely heterogeneous. They're color-coded for you here. Some sense temperature and pain. Others are low-threshold mechanoreceptors that endow you with the sense of touch. And others in red are the proprioceptors. And what they do is they send these tiny processes all over your body and sense the environment and take that information and transmit to the spinal cord and the brain. So if we follow these neuronal endings in the periphery shown here, each also have very distinct targets. I already mentioned proprioceptors in red innervate muscle spindles, while many different nerve endings in your skin innervate things like hair cells or form specialized end organs like Meissner corpuscles. All of them come together to give you this sense of touch. In addition, some of them innervate internal organs, such as the bladder, and we'll talk about this as well. But what my lab was interested in is at the molecular level, how do you sense a gentle breeze or a cactus pricking your finger? We knew that there are these very rapidly signaling molecules called ion channels sitting in the plasma membrane that are directly activated by physical forces such as temperature and mechanical force, but their identity remained unknown for quite a while. So about more than 20 years ago now, David Julius's lab, my lab, and many others identified this specific trip ion channels that are involved in thermosensation. They're actually remotely related to PKD channels that you just heard about. But the ion channels that sense mechanical force have remained a mystery even 15 years ago. So what I'll talk to you about today is summarize how we found these ion channels that we cloned piezos and their role in somatosensation and interoception, which is sensing of internal organs. And then I'll tell you very quickly about how we're finding roles of piezo-1 in physiology beyond the nervous system. And indeed, very appropriate, I'll have some unpublished work about role of piezo-2 in the kidney at the end. So although we were interested in identifying the touch and pain sensors, one of the things in science one needs to do is often ask, what's the most reductionist way to ask a question that get a meaningful answer? Because it was very difficult to identify these channels from neurons, Bertrand Coste, a postdoc in my lab, came to identify such channels and took a very simple approach. And the idea was to find a cell line that you can just grow in a dish that is mechanosensitive, and then to try to identify the channel from that. To do that, he had to do whole cell recording of the cell by doing patch clamp, and with a blunt glass probe, push on the cell. As shown in this video, here's the recording pipette, and here's the probe. By simply touching the cell like this, if the cell has a mechanically activated channel, you could see these currents by simply poking it. So he found out after screening about 30 cell lines that Neuro2A, a cell line commonly used in neuroscience, actually has such an activity. And then by using a variety of bioinformatics and expression analysis, he narrowed down to about 300 genes that could be these potential sensors. And he thought he had it easy now. All he had to do is find which one of these is the sensor. But this took quite a while. So I'm showing this data for you where on the x-axis shows the maximal mechanically activated currents from Neuro2A cells. The top one, this one is in blue, is scrambled control. And then for each of these is a single candidate where he's knocking down one by one and seeing if it affects the current. So to get this kind of error bars, he had to spend about 2 to 3 days on each of these candidates. So after 11 months of getting negative data, he got a very good result with candidate number 72. This was an unknown gene named unknown family 38 member A, so that I don't have to say that so many times in a talk. He quickly renamed it piezo-1 from the Greek piezi, meaning pressure. But this is the data that got Bertrand and I really excited. So the previous results showed that you can get rid of piezo-1 and the cells lose their mechanosensitivity. This shows that if you take a full-length cDNA of piezo and transfect it in naive cells, you get these massive nanoamp level mechanically activated currents. Here's the quantitation. So it's both necessary and sufficient. Piezo-1 has a sister, we call it piezo-2, 40% identical at the amino acid level, and has similar effects when you transfect it in naive cells. Another interesting aspect of piezo-1 was its potential predictive structure. So most ion channels, because they have to sit at the plasma membrane, they traverse the plasma membrane a few times. We call these transmembrane domains. And ion channels have around 2 to 6 of them usually. Piezos had 38. That's more than any other protein in your genome. Many groups, including ours, have solved the structure of piezo, including McKinnon Lab and Zhao Lab, and our collaboration with Andrew Ward. This is the work of Kei Satome. On top of the 38, piezos come together as a trimer, sitting on the plasma membrane, like these giant molecules. So each of these protomers is colored in different color. Only in the C-terminus, the 3 proteins meet each other to form the ion channel. The rest, all these ribbons that you see, are transmembrane domains expanded into the plasma membrane, potentially, presumably, sensing membrane tension. You will immediately notice that they have this very unusual cupping phenotype, and it is current hypothesis that I won't have time to talk about, that any membrane tension will actually flatten this structure and activate and open or gate the channel. But I mainly want to talk to you about is what roles do they play. And after identifying the ion channel, we have used both loss of function and gain of function mutations, both in animal models, mainly mice, as well as from clinical data, to find out what they do. So we were very excited when we found that piezo2 was very robustly expressed in these DRG neurons that we love so much and is responsible for sense of touch and proprioception. So we made different knockouts. I'm not going to name them. The null mutations in mice die at birth, so we use a variety of conditional knockouts to assess physiology and behavior. So one of the first mice that we looked at, this is actually Sanjeev Ranades, one of the first behavioral experiments. He wanted to ask, do these mice sense touch? And we didn't know how to ask the mice. So he came up with this very simple assay that we creatively call tape on the back. So here's a wild-type animal with a big piece of white tape on the back. And as you can see, it doesn't like it. It scratches. And here's a mutant animal. It doesn't seem to care. But the other point I wanted to make is, you see what they give Nobel Prizes these days for? This is a more quantitative assay showing very profound deficit in touch sensation. So this is a little wire that you touch the hind limb of a mouse, which then raises it if it feels it. And in wild-type in gray, you can see as you increase the force, they respond more and more. And you can see the severe deficit in touch sensation. How about proprioception? So this is another model where we're knocking out piezo2 in the caudal part of the animal. And without proprioception, you will be very uncoordinated, and that's what you'll see these mice suffer from. They have complete lack of control of their hind limbs as well as their tail. So for each of these behavioral experiments, we actually go in and do electrophysiology to show that these behaviors are actually caused by lack of mechanotransduction at the nerve endings. And this is work done by Katie Wilkinson, our collaborator, where she's going into these proprioceptors and stretching each single fiber. And you can see in the wild type, she can see massive upregulation of firing when she does this. But in the knockouts, 7 out of 8, so absolutely nothing. So we know this is lack of mechanotransduction. My colleague, friend and collaborator, Alex Chesler with Karsten Bonneman at NIH, then actually identified humans that have mutations in piezo2. So these are actually null mutations, both copies recessively mutated, so there's no protein activity. Has a very interesting clinical background. Obviously, this is a quite rare syndrome. There's less than 50 people worldwide that have it, but many of them were diagnosed as having problems in motor neurons on muscle, simply because nobody in the clinic really thinks of the sensory wing as being important for this. But after they were sequenced and the mutation identified and found that they were piezo mutations similar to the ones that we've described in the mice, they started analyzing and found that they have the exact same phenotype that I described to you, which is lack of touch sensation and proprioception. So this video will show one of these individuals as a test for proprioception. She's asked to walk in a straight line while blindfolded. She's harnessed for her own protection. So this is a task that most of us can do very easily. But as you will see, this individual really struggles because she has complete lack of proprioception. So I'm going to summarize in this slide years and years of work that we've done. But both in mice and humans, in addition to deficits in touch and proprioception, we now know that the piezos are the principal mechanosensors also for a clinically relevant type of pain called tactile allodynia. So this is when touch becomes painful. You might experience this after sunburn, where just wearing a shirt becomes painful, but this is a symptom that many people suffering from neuropathic pain experience. Also, mechanical itch. If you're a kid, remember wearing a wool sweater really was itchy, or a bug crawling on your skin. Rose Hill in the lab, who's here in the audience, showed that piezo1 is responsible for this sensation. But over the years, in collaboration with Steve Lieberlis's lab and Alex Chester's, we've also been very interested in this idea of interoception. So these same types of neurons innervate many internal organs and gather this information, both chemical and mechanical, to regulate physiology. And not only the mechanosensors were not known in many of these instances, the role of mechanosensation in physiology was not known. So we've tried to use the molecules, the piezos, to have these questions answered. For example, both in humans and in mice, there is strong deficit in respiration without piezos. And we've shown that this is because of piezo2 innervation in the lungs. There are many ways to control blood pressure. The neuronal mechanism is called baroreception. So these are sensory nerves that innervate the aortic arch, for example. And if blood pressure increases, there's a negative feedback loop that through the sympathetic nervous system controls blood pressure. And we've shown that this requires both piezo 1 and 2 at the same time. So these senses are not even at the conscious level for you, but some of these interoceptive ones are. So for example, if you have to go to the restroom, you're very aware of this. And this bladder stretch is what controls your timing of to go to the restroom. But again, this is disrupted without piezo2, both in mice and humans. Indeed, the individuals that I talked to you about do not have this sensation, and as they grow up, they are trained to go to the restroom 3 to 4 times a day to avoid accidents. We recently also published that in both, again, humans and mice, gut motility is controlled by piezo2 expression in the sensory neurons. And this is not a direction that's really completely understood. There's so many other questions. For example, one of my postdocs, Kara Marshall, who's at Baylor College of Medicine, is very interested in finding what's the consequence of stomach stretch. So most of research on satiety is based on nutrition, with good cause, of course, but how much this stomach stretch contributes to how much you eat, when you eat, diabetes, obesity is completely unknown, but intuitively when you eat too much, you feel full, which sounds like a very mechanical sensation. So she's very interested in asking this question and trying to find out through piezos how this contributes to physiology and pathophysiology. So till now, I just told you about the role of piezos in somatosensory neuronal sensing. But of course, many other cell types sense, experience, and respond to mechanical force. This includes endothelial cells sensing shear stress, muscles being stretched, many cell types in addition to neurons, including red blood cells that get indented. And my lab and many others over the years have found for the piezo 1 many interesting roles in various aspects of mechanosensation. I won't have time to go through details, but I just thought I'd mention a few of them. So without piezo 1, similar to piezo 2, there's genetics from humans as well as we made mouse models. Blood vessels, specifically lymphatic blood vessels, don't form correctly. And people without piezo 1 are born with lymphedema. And this is interesting because it seems to affect the development of the lymphatic system, mainly valve formation, which are during development areas that experience highest shear force. Others not from my lab has shown that piezos are required for proper bone density formation. And this is quite interesting because there's all this anecdotal information that for example, astronauts who go into outer space come back with reduced bone density. And professional tennis players, for example, have higher bone density in their dominant arm versus the other. And so this is probably one of the mechanisms of sensing pressure and regulating bone density through piezos. And we have done a lot of work on the role of piezos in red and white blood cells. And I'll just mention this quickly in the next 2-3 slides. So the opposite of loss of function of piezo 1 that causes lymphedema, there was also dominant gain of function mutations. So these are mutations that make piezo more active than the wild type. And this causes something called hereditary xerocytosis, which is a red blood cell dehydration phenotype. And it's quite rare, and it's not a very strong symptomatic condition. There's only about less than 200 people who have been identified to have this. But what we have done is make gain of function mouse models for this piezo mutation, and we go back and forth between the human and the mouse condition. So for example, this red blood cell dehydration, we've been able to model in the mice and show that it's specifically because of expression of piezo in red blood cells themselves or their precursors. But because we have conditional control of this gain of function mice, we are able to get very fast mechanistic insight. So another example is that these xerocytosis patients have age-onset iron overload, and we've shown that this also recapitulates in the mouse, but this time it's because of expression of piezo in macrophages. Interestingly, this red blood cell dehydration syndrome, thinking about the sickle cell literature, we thought, oh, maybe this has something to do with protection against malaria. I won't go much into it, but we've shown that this mouse model protects from a murine version of malaria for cerebral malaria. This also let us look if there are alleles of gain of function that are more common from places where malaria is endemic. And we actually found a very subtle gain of function mutation, doesn't cause disease by itself, but it's present in 1 in 3 people of African descent. So again, a very rich genetic source. So coming back to the iron overload issue then, we asked if in the clinic we can find an association with this large population and iron overload. And I'll just show you one piece of data that Shang Ma in the lab gathered. So he got from a blood bank from an African American population, about 300 individuals, and this is just showing their genotype. I already told you we expect 1 in 3 to have this allele. Indeed, in green, we show that the gain of function mutation is present in about 1 in 3, and in orange, we see a few homozygotes. But when Shang asks, if we look at clinically relevant assays of high iron levels, such as transferrin saturation or ferritin levels, and we look at the pie chart, you can see that the carriers of the E756 delete PSO1 mutation have expanded and are responsible more of the pie chart. Interestingly, if you combine these two assays, all 10 individuals that had these high levels, which now becomes clinically relevant, all carry at least one allele of this PSO1 gain of function. So this was obviously a small study. However, it looks pretty promising. If you have E756 delete, you are 12 times more likely to have high ferritin levels. But we realize that larger studies are required. What I want to conclude this section is to say that we're finding, just like sickle cell, interesting clinical data as well as mechanistic data from mouse suggesting that gain of function mutations might have both advantages and disadvantages. The advantage might be from red blood cell deformation, which might protect from malaria. I have to emphasize that this is shown in the mice, but still being investigated in humans. And the disadvantage, at least one of them that we found, is that overactive phagocytosis in macrophages can cause increased iron levels. We are very eager to use these large databases of genotype-phenotype associations such as UK Biobank or all of us from NIH to find out novel areas where PSO1 and mechanotransduction can be important. I want to also say some unpublished work about actually in kidney. So this is the work of Rose Hill, a great postdoctoral fellow in my lab who's here somewhere in the audience. And I really encourage you to go see her talk later this afternoon at 5 p.m. And please talk to her because we are eager to get feedback from this audience. I don't need to introduce this, but everybody knows the renin, angiotensin, aldosterone, the RAS system is essential for fluid homeostasis, salt balance, and blood pressure regulation. And the rate-limiting step of synthesizing and secreting renin from the JG or juxtaglomerular granule cells is well established. But how is this regulated? In response to hypovolemia or salt deprivation, there's very well characterized pathways, including through the sympathetic nervous system or the macula densa in response to low salt to regulate this process. However, we have known for a long time that mechanosensation of the JAG cells themselves might contribute to this. And the idea is that expanded arterioles around these cells might put pressure on these cells, which then would activate a mechanosensor, cause a calcium influx, and it's hypothesized that in a reverse way, this increased calcium would inhibit renin synthesis. However, not only the identity of the mechanosensor here in vivo is not known, the exact role of mechanosensation with respect to the other pathways involved is not elucidated. So Rose wanted to look to see if piezos are expressed and involved in this process. Interestingly, she did not see piezo-1 expression, but piezo-2 is robustly expressed in the glomerulus. And this is on the left, you can see single molecule fish, and on the right, we have a piezo-2 GFP fusion knock-in, so you're looking at the protein expression. And in both cases, we see clear expression of piezo-2, both in JG cells, renin-expressing cells, as well as glomerular mesangial cells. We, of course, want to see genetically if piezo-2 is required for any aspects of kidney function. And to do this, Rose used a variety of Cre lines. One of them is this PDGFR beta Cre ERT2. ERT2 section says that it's inducible, so there should be no developmental consequences. She induces the knockout in the adult. And this Cre would be expressed in a variety of specialized vascular smooth muscle cells, including JG cells and mesangial cells. Rose has also shown exact same result with the renin Cre mice, so we think this is in the combination. None of the Cres are perfect by themselves, but the combination suggests the involvement of these cells that I described to you. And in its normal conditions, she can already see a significant increase of plasma renin, as you can see on the left. She also wanted to test these mice in a challenge condition, and she induces hypovolemia by PEG, and she sees a much more robust induction of renin expression, in addition to some of the downstream pathway, including angiotensin II and aldosterone. Rose has started also looking at downstream consequences of this, and remarkably finds that in these mice, both in the PDGF as well as the renin mice, she sees a robust activation or increased glomerular filtration rate, which is very exciting. So you can go and see more data with Rose's thing. I just wanted to give you a quick overview of what she's doing and finding. But to conclude this section, I would say Rose thinks that piezo-2 in renin-expressing cells regulates plasma renin by acting as a brake on the RAS activation pathway. Interestingly, for downstream consequences, she finds not just upregulation of GFR, but also, which I'm not showing you, systemic blood pressure. What's really coolest thing about this, though, I think, is that this suggests that the same molecules that we sense to sense touch is actually functioning in the kidney to sense pressure. In that vein, I wanted to end with telling you that piezos are actually not just present in vertebrates, but are conserved across many different species. They're in animals such as invertebrates, C. elegans, and flies, unicellular organisms, also in plants. So plants also sense mechanical force in many different ways. One of them is the roots of plants. As they get into the soil, they have to, for example, sense the soil that they have to penetrate and decide if it's hard enough to go through it or go around it. Ali Reza Musavi, a postdoc in my lab, showed that piezo is expressed robustly at the root tips of this plant called Arabidopsis thaliana. And data that I won't show you, but just mention that if you knock out piezo in plants, you see pretty severe phenotype in the ability of the roots of these plants to penetrate hard soil. So we're allowing these plants to penetrate an agarose gel, and in the left, the wild type can easily penetrate. You don't see the roots because of the focal plane. However, in the mutants, they cannot penetrate and go around it, and you can very easily see the roots there. So again, evolutionarily, it seems like once this protein as a membrane tension pressure sensing has been evolved, evolution uses it again and again for us to sense touch, to sense pressure in the kidney, or for plants to sense hardness of the soil in their environment. I just wanted to end by thanking current lab members, alumni, and collaborators. In blue, I've highlighted some of the people who've done the work that I described today. Special thanks again to Bertrand Coste, who cloned piezo in the lab. Sanjeev Ranade, with many different postdocs and students, led the work on the somatosensation and interoception. Shangma did the work on the piezo-1 gain of function I talked about. Ali Reza I just mentioned in the plants, and especially Rose again started this work on kidney, and our collaborators Alex Chester and Jeff Miner and Stuart Shanklin, who are helping us understand the kidney and work on this. And I also wanted to end by showing this wonderful picture that we took last night at dinner. So Jeffrey Miner, who's of course comes to this meeting all the time, he was in the same graduate school lab that I was. He was a senior student when I started, and he was actually my mentor, so it's wonderful to see him here and interact with him again. And Rose is also here, who's the next generation. And one of the things I wanted to say is I really appreciate this invitation. It's great that you guys have a broad perspective and invite people outside the field. And I think one of the great things, I was a molecular biologist that got into the touch and pain field, and I think being from the outside really helped me look at the question in a different way. So everybody was trying to clone, these mechanosensors from neurons, and my molecular training took us this very reductionist approach of just finding in a cell line. So I think this cross-fertilization of fields is very important, and Rose is on the job market and wants to focus on kidney biology, so I hope you welcome her with open arms. Thank you for your attention.